Welcome here. So what we would like to do is discuss with you the potential of gene editing, new methods of gene editing, and the potential what it might have for therapy which is developing. I think the potential is huge. So what we're going to do is I will give you a brief overview of the technology and then have three experts addressing various aspects of how they think this will develop and what the state of the art is in a specific field. So if you go back for gene editing historically, it started something in the 70s by taking a zygote, an embryo of a mouse, introducing virus or DNA into that, and generating what was then called the transgenic mouse. So the issue here was, so there was a new Mendelianly inherited sequence, but these transgenes are randomly integrated. There was no way to predict where they would go. And this changed in the 80s when homologous recombination was invented, where you could now design and predetermine what you want to change in the genome. And this led to really an explosion in biology. And this will be the outline how animals with a knockout were made. You have to do this in embryonic stem cells, importantly. You have to inject those into blasts, make what's called a chimera. You have to cross a chimera and hope that you get germline integration. And then you have a mouse which is a predetermined mutation, which is really was quite remarkable. So it works very well in mice, but mice were at this point the only species where the embryonic stem cells, and you need that. It's very time-consuming. You have to go through generation of mice. And as I said before, it's applicable only to mice because no other species had robust embryonic stem cells which could integrate. So that really restricted it, and it didn't work in human ES cells. So this all changed with the new gene editing tools, which are either proteins. I will not talk about the talons, the zinc fingers, mainly about the CRISPR, where an RNA guides the Cas9, which is protein, to a predetermined position in the genome and generates a double-strand break, and this could be then used to edit the gene. And the question was, how efficient is that? And so I give you one example how efficient that is, which was to us certainly very amazing, was to ask the question, how many genes can you, in an ES cell, can you target at the same time? And so in this case, my lab did 5 genes. 5 guide RNAs with Cas9 to different genes, and it doesn't matter what the genes were. And it was really remarkable. 50% of the clones had all 5 genes, both copies on, one was on the Y chromosome, really mutated. That was incredible, efficient. But really you had to go through this whole thing making chimeras, and so the question is, could you do it in the embryo? And you can. You just inject the RNA for Cas9 or for the guide RNA, which guides it to where you want to go. In this case, 2 different guide RNAs to make double mutations, and the efficiency was 80% of the pups you got had mutated both alleles of both genes. So that was incredibly rapid. And then you could ask the question, can you, and that's important for what we're going to talk about, can you make predetermined mutations, where you introduce a predetermined mutation into a gene, and in this case, again, with 2 genes, and you have to just co-inject a DNA which has the mutation, and you get with a 60% efficiency in these point mutations in both genes. So this was a very remarkable efficiency of this technology. So let me just summarize that. So you inject guide RNA, Cas9, a piece of DNA, into a zygote, an embryo, a fertilized embryo, make a blastocyst, and make a mouse. So the whole thing is really efficient and rapid, 3 weeks. And you can't make this shorter because that's the gestation time of the mouse. So it's really quite incredible. So this technology, editing really has enormous influence on disease modeling. We can now make monkeys which have predetermined mutations, which is being done. You can make it very efficient in human ES cells. You can use targeted gene therapy, and we'll hear about this, either in vivo, editing the genome in vivo, or combined with cell therapy. And of course, because it's so efficient, because it works in animals, the big question was, should you do it? Can you do it in human embryos? What is the prospect of manipulating the human germline? And this has been rather, of course, a big discussion. There has been a National Academy of Science committee which worked out guidelines, and 2 of us were on this committee. So this is something which is a discussion is ongoing. Now let me raise one final issue. This paper came out about 2 weeks ago, and it addresses the issue of off-target insertions. And some of it went through the press, was discussed. It's a very short paper, and let me just give you the key conclusions. It was based on mice, not on cancer cells, which are often used, so that is a good thing. And they argued they got 1,800 off-target mutation, and 40 to 50 were in coding regions. Now, this is really disconcerting. But the other thing they argued was there was no obvious homology of the guide RNA with the mutated sequences. So we are really at a loss, according to this paper, to really predict what's happening. Now, this paper is controversial. So the companies who are using CRISPR-Cas raised a lot of criticism because their stock went down and so on and so forth. So I think it's really an issue which is unresolved, and I believe we may address the issues here, and probably we'll hear this later in the talk. So there are 3 experts who will talk about. Luigi Naldini is the first one from Milan. He's really an expert in gene editing in human hematopoietic cells. Some of these issues have gone successfully already in the clinic using various manipulations. Alessandra Biffi from Harvard will talk about, or will address gene editing and gene therapy from a clinical perspective. So where are we? And finally, Katriona Jamieson from UCSD really will talk about issues again, how can we affect complex human diseases. So with this, I would ask now Luigi Naldini to lead his discussion.
Okay, good morning. So I will briefly discuss the potential of gene editing when manipulating hematopoietic stem cell. Now, obviously, stem cell-based gene therapy really relies on the year-long experience of transplantation of stem cell from healthy donor. The difference being that the cells are manipulated before infusion by gene transfer or gene editing, and the cells come from the same patient. So essentially, it's an autologous procedure, which has a lot of advantage because there is no immune barrier to cross. And this is really an important way to make the process safer as compared to using donor-derived cell. The cells are then infused into the patient upon conditioning, which makes space in the marrow so they can establish a permanent population of gene-modified stem cell which can feed the different lineages of blood. And if the cell carry a genetic changes which correct the disease, this change will be passed on to the progeny. So we can treat a variety of immunohematological disease, storage disease affecting peripheral organs or brain, and potentially as well cancer and infection by modifying the cell properties. Now, the challenge for this strategy has long been to achieve efficient genetic modification of stem cell while manipulating them ex vivo without perturbing their function. These are very sensitive cells. They may apoptose, they may differentiate, and they may lose the capacity to engraft. And also to achieve enough cell when you treat them to treat a patient. Usually in clinic, of course, you can't rely on very few modified cells to have a safe recovery of the patient upon conditioning. We also need to be able to regulate gene effectively and physiologically, and any type of genetic modification of stem cell is a mutagenesis by definition because we change the DNA. Otherwise, it would not pass to the progeny. And this has some risk, inherent risk. If we use gene transfer, this may be due to insertion of the vector, which is random, as Dr. Jenic mentioned before, so you can control that, and you may activate occasionally a cancer gene. If you use gene editing, you may have a non-target defined region, but you may have off-target, which may, again, inactivate and inadvertently tumor suppressor gene or cause translocation, which may activate. So that's the type of scenario we have to be aware of. Now, despite the challenges, hematopoietic stem cell gene therapy has made a long progress. We have several established treatments now, one of which is the first gene therapy using hematopoietic stem cell. this is using gene transfer, so conventional gene transfer, is now a product on the market. So this can be essentially prescribed to patients going to the clinic and having their own cell modified for gene therapy. We have then an expanded use of this approach using lentiviral vector technology. There's several trials over the world ongoing. More than 200 patients have been treated today. Alessandra Biffi will mention that in more detail, I guess. And these have shown remarkable efficacy and safety today. So that's an important advances against which we have to compare where gene editing will go versus this being traditional gene transfer. And the disease which have been treated and under different trials, some of which have been completed, and some of these therapies are now being filed for registration and to get to the market are, again, immunodeficiencies, blood disease like thalassemia and sickle cell, and storage disease like leukodystrophies, which are neurodegenerative disease. So we may see that in the future, autologous stem cell gene therapy may become a preferred option for the many patients that today would undergo an allogeneic transplant. And this could save important risk to the patient because of the autologous nature of the cell, and potentially because we may diminish the harshness of the conditioning, because we don't need to completely repopulate the hematopoietic system with the allogenic cell. We can only establish, in some cases, a fraction of normal cell. Where the field is going to make this even better and safer, we are trying to increase the number of stem cells that we infuse by potentially expanding them, and this will make transplant even safer because it will be faster reconstitution. We are trying to move away from conditioning which use chemotherapy, which are very toxic to the patient. In the long run, they may suffer, of course, from long-term tissue damage, and we are looking at more precise engineering, which is gene editing, as mentioned, in which we target specifically our modification to the desired sequence. There's two advantages of using gene editing. One, we may inactivate gene selectively, in which case we can modify a cell response, for instance, to a virus, make it resistant, or we may correct the mutation in situ, right where they are found in the genome. So this application, I think gene disruption, as I said, is unique potential, is highly efficient. We are getting, as Dr. Jenic mentioned, very high efficiency, even in primary cell for clinical use. And these have been developed and is in early testing in the clinic for potentially make a stem cell and a progeny resistance to a virus by inactivating the receptor for that virus, or to reactivate some like fetal hemoglobin in case of thalassemia to compensate for a defective gene. More difficult is actually the gene correction, which relies on homologous recombination. This is more difficult because the manipulation requires more complex. The advantage is we restore function and expression control. So the actual endogenous gene is, in fact, corrected in situ. And as mentioned, the issue is really balancing how we can achieve this efficiently versus what are the risks of mutagenesis on the genome. The advantage is that using nucleases, we can always target only a fraction of the genome. This fraction may comprise of target, which have to be defined, but it may not be as huge as the whole genome when we have a randomly integrating vector. Because today we are perfecting the technology, we are seeing improved efficacy and specificity, but we still, in the case of hematopoietic stem cell, we have a limited yield of cell bearing the correction. It's conceivable that the first application in humans may exploit disease in which we don't need to have 100% gene correction, and only few corrected cells may be sufficient to rescue the disease. And that is our work, which we have been discussing at this meeting, in which we essentially have modeled gene editing through hematopoietic stem cell in immunodeficiency, which is a SCID X1, and have been able to show that in a mice model, we can have a full and safe rescue of the disease, even if we have only 10% of gene-corrected cell in our cell product. And we have been able to show that using optimized protocol and technology, either based on Zinfinger or CRISPR-Cas reagents, we can safely and reproducibly achieve those levels in human cells. So that would set the stage for what could be the first clinical testing of this homologous recombination-based correction in humans. Thank you.
Okay, thank you.
Good morning. As mentioned by Rudolf at the beginning, my job today is just to give you some clinical sense of where gene editing strategies could provide a therapeutic indication, and in particular to set the indication in the context of currently applied gene therapy approaches, gene transfer approaches. I really don't need to tell you in detail more than what Luigi has just done, the differences of these two approaches, but I think it's important to underline that on one side, gene transfer has a robust history of clinical testing, and this history is dating to the early 90s and has resulted in the registration of the first product in 2016 and is moving into new registered product in the upcoming years. Whereas on the genome editing side, we are right now approaching clinical testing. So the important part of the talk I'm going to give you today is really defining where is really the right setting in terms of clinical indication for testing these novel strategies in humans. So the first indication that I would like to discuss with you is that of primary immunodeficiencies that has been already introduced by Luigi. This is also the first indication in which the traditional gene therapy based on gene transfer has been applied, and where we have probably the most robust clinical data with an overall huge number of patients who have been treated. Obviously, the numbers are related to the fact that we are in the field of rare diseases, so we are not discussing about the numbers that you would expect to find in more common diseases like cancer or neurodegenerative disorders of adulthood. But being in the field of rare, more than 100 patients with immunodeficiencies have been treated by gene therapy and gene transfer with a remarkable degree of success. Survival is definitely very high, as well as the response rate that is above 80%. And these numbers are particularly relevant whenever you refer them to the experience with the donor hematopoietic cell transplant, which is a standard of care for these kind of patients that has definitely still right now a high morbidity and a high mortality with partial response in the most severe cases. And this is also the setting where the first hematopoietic stem cell gene therapy approach based on autologous stem cells, as just mentioned, has been registered as a drug, and this is the experience for ADA-SCID run in Milano under the sponsorship of GSK. Why primary immunodeficiencies are so relevant in terms of setting where to test these novel strategies. It was already alluded before. The main reason is that in this condition, the gene-corrected or transduced hematopoietic stem cells, and most importantly, their mature T progeny, T cell progeny, would have an in vivo selective advantage. So because of their gene correction, they would expand naturally in vivo over the non-corrected ones. So in this setting, we could anticipate to achieve clinical benefit also if we infuse only a little fraction of the pool of the transplanted hematopoietic stem cells that has been sufficiently corrected and successfully corrected. This little fraction, this 5 or 10% of the gene-edited cells, would have the chance in vivo to expand and deliver a therapeutic outcome in terms of phenotypic correction in the affected lineages. The other important consideration that would render important to test gene editing in this setting is that physiological regulation of the expression of genes involved in hematopoietic stem cell function, proliferation, and T cell function is extremely important. And this is told by the clinical experience in the early days gene transfer with gamma retroviral vectors. Obviously, gene editing and correction of mutations in the affected genes allows us to achieve this physiological regulation of the corrected genes that otherwise cannot be achieved by gene transfer, where the corrected gene would be expressed by an exogenous promoter in the majority of the cases. Again, also this was mentioned, the other area where gene transfer has been applied with remarkable clinical outcomes in terms of independence from transfusion or reduction of the transfusion rate is the area of hemoglobinopathies, both for beta thalassemia and for sickle cell disease. And the results are being reported over the past few years in the press. This setting offers a very new and interesting area for the application of genome editing, that is really the option of regulation. As mentioned, here we can really exploit a physiological regulation of the expression of the globin chain, which is offered by BC11A, that was identified originally by Stu Orking, and work on that in order to induce in the patients affected by either sickle cell disease or beta thalassemia the production of fetal hemoglobin that would overcome the affected and abnormal hemoglobin the patients produce and determine therapeutic benefit. So this is a totally different approach that is really being brought to the clinics in these days by Sangamo. The third example is that of lysosomal storage diseases and leukodystrophies, so diseases of childhood that affect mostly the central nervous system of patients. These children usually have a very severe disease manifestations. After a few years of normal life, they start losing their motor and cognitive skills, and these are conditions that usually have no treatments available. This is also a kind of condition where hematopoietic stem cell gene therapy based on gene transfer has demonstrated a robust benefit, and in particular, both in the experience of metachromatic leukodystrophy and that of adrenoleukodystrophy, a remarkable benefit was observed in the patients who have been treated by HSC gene therapy in the form of prevention of their motor and cognitive deterioration, and the children have experienced, if treated sufficiently early in the disease course, they have experienced a normal motor and cognitive evolution. However, what was pretty remarkable and striking in this setting is that benefit was strongly dependent on the levels of therapeutic gene expression. So whenever benefit was observed, it was in the presence of a robust expression of the therapeutic gene well above the normal levels of expression. So can we do this by gene editing? If you think about what we have just discussed, in situ correction of mutation, it's difficult to think that we could express the gene that has been corrected above the normal because it would be simply regulated by its own promoter. So this is definitely a challenge for gene editing that was overcome by gene transfer because of the use of exogenous promoters and multi-copy gene transfer. So the solution in this setting, in order to render this novel technology applicable for these diseases, is to add the therapeutic gene of interest in a specific locus that would have the possibility to express the gene at high levels. And this was obtained at the level of the liver for an in vivo gene therapy application for lysosomal disorders, using as a target locus the albumin locus that is a locus highly expressed by the liver cells. This kind of approach is, again, entering the clinics right now, and on the other side, the use of hematopoietic stem cells is moving forwards clinical development thanks to the use of candidate loci that in this case are highly expressed in the context of the hematopoietic lineage, and in particular myeloid cells that are the relevant population for treating these kind of diseases. Finally, the important application of gene therapy that has been successfully reported recently is that of cancer, and in particular CAR T cells. As you know, this is an exploding field and is mostly based on integrating vector, gamma, retro, and lentiviral vectors that deliver a CAR into the T cells of the patients. Also in this case, despite the extremely successful clinical results, gene editing may have an important space and may play a critical role, as recently shown by the group of Michel Sadelain, who clearly shown how editing of the CAR receptor at a specific locus, a sort of physiological locus, allows to improve dramatically, at least in mice, the results of CAR T-cell therapy. And this is another important application that will likely enter the clinic soon.
Good morning. I'm Katrina Jameson from UC San Diego. I'm the director of the Serum Alpha Stem Cell Clinic there. So I've certainly been able to witness the benefit of gene therapy in the stem cell realm through our Alpha Stem Cell Clinic network. So it's been very exciting. However, in my lab, we've studied our endogenous gene editing systems to say, is there something that would limit the efficacy of gene therapy approaches that involve lentiviral vectors? And do we have endogenous gene or transcript editing systems that actually evolved to protect us from lentiviral or retroviral integration? So we've been working on what are called ADAR editases for quite a while. These are adenosine deaminase associated with RNA editases, highly conserved. They were first discovered in trypanosomes as a way to introduce transcriptomic and subsequently proteomic diversity. Most recently, the cover of Cell showed that actually octopus, cuttlefish, use these to increase their ability to adapt to the environment and learn, so they have more neuronal plasticity as a result of activation of ADARs. And in humans, we use these enzymes more to rewrite, recode, and rewire our transcriptomes, or in other words, our RNA. So usually when you hear about DNA editing, you think about therapeutic benefit, gene editing. What we're talking about is RNA, and that this is a protective mechanism for our stem cells to protect themselves from retroviral integration. So we have 3 versions of ADAR. The one that we've studied most is ADAR1, and there's a nuclear component called P110 and then a cytoplasmic P150. And what they do is they deaminate adenosine to inosine that's subsequently read as aguanosine. So the question is, why is that important? Well, these ADAR editases can introduce new splice acceptor sites. They can change the coding sequence and change RNA shape. And they act in the context of primate-specific ALU sequences. So this is what we were taught was junk DNA, don't worry about it. However, it represents 10% of our genome, and this is primarily where ADARs act. They also act to edit microRNAs, long non-coding RNAs, and 3' untranslated regions. So they're very, very strong effectors of the transcriptional output of what the gene is trying to say to the cell. Now, ADARs are part of an antiviral response mechanism activated by inflammatory mediators like interferon gamma, and as I mentioned, they really evolved to protect our cells from retroviral integration. So you can imagine as we use these vectors for gene therapy, it may limit the effectiveness there. So what we started to notice, along with many others, is that these ADARs are activated as pre-malignant cells decide to become malignant. And the reason that's so important is because they tell that cell to clone itself. So that cloning or self-renewal capacity is something that should only exist in a stem cell population. But this occurs a little further down at the level of the progenitor, because in leukemias like chronic myeloid leukemia, that progenitor has increased interferon gamma receptor expression as well as IL-3 receptor alpha expression. These both signal through JAK2 to activate transcription of ADAR. An oncogene called BCR-APRIL does the same thing, and what this does is it edits a microRNA that's normally responsible for differentiation, and because it edits, it can't mature and it can't work. So basically, the reason, again, this is important is we can use this to predict progression. Gordon Mills and colleagues published in 6,500 patients, 17 different tumor types, the same enzyme was activated. So clearly it's an amplifying event. It causes progression, but we think we can come at it either by developing very selective ADAR inhibitors, which we're collaborating with GSK to do now, or by coming in with a JAK-STAT inhibitor that dials down activity of ADAR. So we're quite encouraged by this. The main thing is just being able to detect it. And there's another gene editing system that actually edits DNA, also antiviral, and this is the APOBEC family of deaminases that have been found to be vitally important in cancer evolution and a source of enzymatic mutagenesis in breast cancer, as originally published by Rubin Harris and subsequently by Mike Stratton, to show that the APOBECs, when activated, actually impair genomic stability and are really vital, again, for progression because of this C to U change that is subsequently read as a T. So when you look at the number of cancers that have activated these enzymes, it's really quite astounding. So I think that we need to look at our endogenous gene editing systems to see when they're activated, when it's appropriate, and how can we turn them off. So in summary, we've really been looking at healthy human aging, and I do think that exists, as opposed to malignant reprogramming of progenitors into cancer stem cell populations. And what distinguishes benign normal aging is that the aged hematopoietic stem and progenitors actually have a slightly reduced self-renewal and survival capacity with age, which is actually a good thing, because if you develop a mutation in that progenitor, it's less likely to survive, as opposed to what happens with ADAR overexpression. That progenitor learns to clone itself, particularly in the context of splicing that promotes pro-survival BCL2 family member expression. So we think that a targeted approach to inhibiting ADAR can actually reduce cancer stem cell evolution, and certainly something that we'd like to work on more and potentially clinically. Thank you.
Okay, so you saw there was a lot of excitement, a lot of interesting issues coming up for application. And let me ask you for any questions which you might have.
My name is Antonio Regalado. I'm a reporter at MIT Technology Review. Maybe we can talk a little bit about the off-targets. That paper's been heavily criticized, but it did remind people that these off-targets exist. So I guess in your presentations you didn't discuss the possibility of off-target effects in the cells that you're editing. So maybe you could tell us what you're seeing and the level of concern and how many off-targets would be required to cause a problem.
This is obviously a huge problem, and the FDA is very concerned about, and I think the evidence is rather complex. So maybe, Luigi, if you want to give your...
Yeah, I mean, obviously this has been always an issue in the editing field, and there has been effort to develop methods to identify off-target, which are preferably non-biased. I mean, the obvious way would be to look for in the genome sequence which have some level of homology to the intended target, to screen activity at those sites. And this has always shown to be an imperfect approach, because we can't fully predict which type of homology is relevant or not according to individual reagents. So the preferable approach is to use unbiased way, essentially looking at where the breaks are. And then breaks would be, of course, indication of activity of the nucleases, and then we could deeply sequence genome of treated cell to look at presence of non-homologous enjoining at those breaks. So this is essentially the way we go. The limitation of that approach is the sensitivity of deep sequencing. Essentially, we can't really go below 0.1% of mutation at this individual locus, which means that there is a whole potential fraction of off-target activity which would escape detection below 0.1%. So you would have a major difference between on-target activity, which can be sometimes easily 50% of the allele, 100% of the allele being modified, where the off-target would be less than 0.01%. So of course, there's 4 log differences. However, if we are infusing 10 to the 8 cell in a patient, that makes a lot of space for those off-target sites to be present. And if these are at potentially dangerous genes, they may still select out. So the approach has been essentially to try to, as comprehensively as possible, identify potential off-target, screen for them, and essentially also rely on some sort of biological testing. So your cell product is going to be tested in assay, preclinical model, for any sign of abnormal growth or transformation of the cell, which ex vivo can be performed relatively easy. So and then finally, the last point is off-target are not so much platform dependent, but they are really dependent on any individual reagents. So every reagent, whether it's a Zinfinger, Talent, or CRISPR-Cas reagent, has to be individually assessed for its own target, and there is a range of target activity. So it has to be product-specific definition, and also context-specific, because in some disease and in some application like T-cell, there is less concern, for instance, based on previous study, that even if you have some low level of target in your product, this may be well tolerated. Other targets like stem cell may call for more prudent approach and so more careful analysis. So that's the way we are now. I mean, we don't have a solution yet. We have to continuously investigate the issue by a combination of approaches.
Can I just ask you to address one further question?
Yes.
That's very clear. How much off targets do you actually see in the cells that you edit, and do you see these abnormal growth? So tell me, you told me the process for the...
Okay, so in most of the products which are brought to clinical testing, there is no more detectable off target. Essentially, the off-target are below this detection level. That's what we have seen. So in our CD34 cell in the preclinical studies, by improving the reagents, we have completely eliminated the detection of those targets. As I said, they could be still there at below detection level. And I think that would be the acceptable approach. Essentially, you would want to be at that level at least, not detecting it.
Okay, thank you.
And a related question, I didn't follow whether in your search for off-target effects you were using the algorithms that suggest...
No, we are using the unbiased approach always. There's many of them being reported. Some are more sensitive than others. So essentially, you really go first to make a list of this by unbiased strategy, and then each of them are deep sequence, not only in an indicator cell, but also in your product itself, so the cell which has been exposed.
Okay, sure.
Maybe a question for Alessandra. That was a very interesting chart that you showed about the number of patients treated with gene therapy. It's a small number. So why does it make sense that this should become a product by a company, not a hospital-based procedure as it is now? The small number of patients, the nature of the treatment. Why is this a company product at all?
So I think the matter is really accessibility. Hospital-based means clinical trials and means dedicated funding and dedicated and specific clinical question. A product is something that, first of all, was demonstrated already to be safe and feasible according to parameters that are to be defined in the context of the clinical trials that were run before registration in order to bring the product to that level. So it's a medicine for which we already have a defined labeling in terms of safety profile and expected outcome. And this is the end point, let's say, of every clinical development and preclinical development of new indications. So to get there means that we have already demonstrated this and means that the patients could have access to it independently from the ability of researchers to work on that product with additional questions or with additional funding. It becomes a drug available to every patient who's diagnosed. So they don't need to search for the trial. They have a diagnosis and they have access to the treatment, independently from the rarity of their disease.
I wanted to add to that. So in terms of, this is Katrina Jameson again, in terms of gene therapy approaches, the reason that it's so important to have it as a product is because there are so many moving parts, reagents that have to be used, as Dr. Naldini was mentioning, in exact sequence. It's not the same as when I used to do bone marrow transplantation where you process the cells without gene manipulation. This is something that has to go through GMP manufacturing, good manufacturing in other words, to ensure that the process has not changed and is very rigorous. And that's absolutely essential to prevent any untoward effects.
So the last consideration is, of course, this is also to be seen in terms of platform development, because obviously the genetic disease we are discussing are the testing ground for this technology, where there is a more obvious readout whether they work or not safely, but then you can see an expansion both to more common genetic disease like the blood. I mean, thalassemia is relatively prevalent, so in that case, there will be, for a product, much more a market, and then, of course, expanding even to cancer by different strategies. So I can see also an investment in terms of commercialization of the platform of autologous cell manipulation.
Maybe I could add one aspect too. The issue is the genetic alteration of the genome. It's irreversible once you have done it. and it has potentially other effects which we have to exclude. But you can do also something else. You can edit the RNA, which would be not genetically, or you can edit the epigenome, which is really now has been shown to work, and Dr. Naldini's laboratory worked on it, our lab worked on it, as many others did. So this is in principle works, and it's reversible. So for genes which are silenced because of whatever epigenetic, there are imprinting diseases, there are other diseases, you could reverse that by editing the genome, but it would not be irreversible. So that's another toolbox which is being developed.
I talked about RNA editing, just to follow up on Dr. Janish's point, more as a potential limitation for viral vector-driven gene therapy, but in actuality, as Dr. Janish is mentioning, it could be used to make cells potentially impervious to HIV infection. If you think about ADARs as our natural antiviral response, really they evolved to protect us from viral vectors or viral infection such as HIV. So what happens if HIV infects a cell, that triggers ADAR, adenosine deaminase associated with RNA, to basically behave as a sushi chef for that viral integrant or prevent proviral integration. And the other thing it does, as shown by Stu Orkin and our lab and others, is it expands the stem cell population. So now you have a self-renewing expanded stem cell population that's impervious potentially to HIV infection. So it could actually be quite useful therapeutically in the future.
Any other? Yeah?
As long as we have the experts here, some of these gene therapies involving CRISPR-Cas9, I think that they introduced the DNA for Cas9 into the cell, or that would be the concept. Do people have thoughts about what happens on the rare occasion when this DNA integrates into the genome?
I think there's always a possibility, although some of these vectors will have very low efficiency, but they could integrate. Any DNA you put in the cell has a potential to integrate and does something unpredicted. I mean, this is just how it is, so that's why it's useful not to use DNA. And that is, of course, the most useful thing, but of course, for gene correction, you need a piece of DNA to do. So this is always an issue, which one has to address by what we heard.
In the ex vivo strategies, now most of the nucleases are delivered as protein or RNA. So this would take away. Of course, you still need the DNA for the template, for the donor, but that would not carry the nucleases. So the ex vivo part, I think, is mostly solved, this issue. The in vivo approach still rely today mostly on vector which express the nucleases. Although there is emerging data, which are, I think, quite interesting, of using nanoparticle that would again deliver only the RNA or the protein for the nucleases. So all of this, because essentially we want to have a transient expression and no risk of integration.
But what would be the risk if you, as a medical person, saw Cas9 integrated into someone's cell and being expressed? Would that be alarming?
So Cas9, I think, has a great advantage because it's really become a nuclease only if it is loaded with the tracer, the CRISPR RNA. So the beauty of that enzyme is that in the absence of the CRISPR, it doesn't even bind the DNA. So essentially, it's really a regulated one. So if you only express the Cas9, presumably in the absence of any tracer-like RNA, you would not have potential risk of a random or targeted nucleases. The only issue I would raise in vivo would be a potential antigen being expressed in the cell, which could be targeted by the immune system because, of course, Cas is derived from bacterial. So you may not want to have that.
I mean, mice have been made which express Cas9, and so far there hasn't been seen some phenotype. I mean, but this is, of course, a rather not a very sensitive assay. So coming back to what we heard, the really successful gene therapies, or the most useful one, was to add a gene, right? To add a gene to correct something where the gene is defective or not. And that has been started, a long history has been started in the 90s. And gene therapy, I think, got a rather bad reputation because of some of these early trials. And maybe it might be worthwhile to revisit this, what happened there and how this was resolved, because the most successful clinical application were really just taking a virus into the Nalini laboratory really has pioneered some of those applications, and how to avoid this insertional mutagenesis.
Yeah, I think it's a learning curve, right? So, I mean, the early generation vector were less efficient. They were based on gamma retroviral vector, less efficient and more dangerous. And we have seen that in the clinic because obviously, despite the early success in the patient, there has been emerging leukemia, which were linked to insertional mutagenesis. These, of course, have slowed down or even arrested the field for several years, and essentially scientists went back to the bench to address this issue, and I think we came up with improved vector. Essentially, lentiviral vector are, if you want, an improved version of the early gamma retroviral vector, mostly for ex vivo therapies, as much as AAV has been developed as an improved version of adenoviral vector, which was toxic in early trials. So I think the current success of gene therapy is really the outcome of hard work at improving the tools and making them safer and more efficient as compared to the early one. Most likely, gene editing will undergo a similar curve, learning curve, where early tools will be perfected, but you need testing. So I think the important point is that you can't keep working in the lab to have the perfect tool and never test in clinic. So there must be some virtuous testing and improvement.
And if I can add 2 comments. First one, clearly many of the concerns that have been observed in the clinics were not seen in the mice, so not necessarily the tool would be informative in every instance. And the other comment is that even in the setting where the first severe adverse events in gene therapy were observed, which are immunodeficiencies, the outcome in that setting of the overall gene therapy treated patients is much better in the presence of the adverse event as compared to the allogeneic stem cell transplant, which is what these patients have available. I've shown you, considering including the number of patients who experience a severe adverse event, I've shown you a survival which is above 90%. If you look at survival of PID patients receiving allotransplant, is significantly below that number, and in any case, not every PID patient would have a donor. Usually, allotransplant is not indicated in these children, so even those who have a donor would have a survival below 90%. So overall, even in the presence of SAEs, gene therapy experience has been extremely successful and with a rate of events that is at least comparable, if not lower, to other standard of care measures.
So maybe you can raise another issue. Now we talked about really ex vivo gene therapy. That's what really most successful, because we can do this. We can have quality control to a certain extent before we do this. But about in vivo gene therapy, and it was, I think, Dr. We mentioned this, in liver is a good target, and there are clinical trials ongoing to correct hemophilia, and it looks, I think, very good. But gene delivery, of course, is the key thing. And I just want to raise a question from you. How successful are we to deliver genes to non-peripheral tissues, like to brain tissues? Is there any, so maybe we can just briefly mention that, where we are.
Yeah, I think in vivo gene therapy, as you said, equally relevant to be mentioned. Gene therapy mediated by adeno-associated viral vectors to the eye is another field of great success. It's obviously one of the indications where there's a perspective for marketing registration that, again, we underline because we think is fundamental for moving ahead the field. And the same is true for in vivo gene therapy directed to the brain. Again, adeno-associated vectors seems to be the ones working best. There are many serotypes. The most advanced is probably serotype 9 that can target the brain efficiently, and the central nervous system in general efficiency, even when injected intravenously. And this is the experience, for example, of the spinal muscular atrophy trial that has demonstrated a remarkable benefit in children who have a devastating disease, and is now moving into phase 2 testing, extending indication also to the less severe forms, so spinal muscular atrophy type 1 and type 2 with different routes of delivery. So there's an exciting phase for gene therapy that also includes in vivo delivery of vectors and editing tools.
Maybe I can add on the liver part, where of course there is amazing result from AAV again based gene delivery, mostly in hemophilia, as Dr. Jenisch mentioned. I think we are now more than 9 years out for the treated patients, which show stable expression of factor 9, mostly because there are some trials in factor 8 very recent. And this is essentially one intravenous injection. It's essentially an outpatient treatment, and they are essentially treated of their disease complication for 9 years now, and they still see stable activity. So that's a remarkable benefit. This is still a non-integrating system. We still don't know the molecular detail. Editing may also do a similar approach, but there the challenge again is to compare efficacy and safety of the two platforms, because just gene addition works so well in reality that you have to see whether the benefits of gene editing are real or not for that disease.
Yeah.
Just mention, going back to your question about why are there so few patients, relatively speaking, compared to bone marrow transplantation that have been candidates or benefited from gene therapy, and that's because we need a stem cell gene therapy network with centers of excellence where patients can clearly identify where they can have this therapy done safely. And that process is something that the California Institute for Regenerative Medicine has essentially been building by setting up the alpha stem cell clinics. So we have accelerated clinical trial agreements, confidentiality agreements, because as we talked about, this often involves different companies. We have sent an IRB, an institution review board, where if it opens in our site, then it's accepted by the other sites. So this is UCLA, UC Irvine, City of Hope, UC San Diego. And because it's been an acceptable or actually quite successful paradigm over the last 2 years, it's expanding to 2 more sites in California. So I think if we can really make this templated, we talked about the process before for how you make the cells. Now we need to deliver properly to patients, so it's far more accessible and safer, because we have a cadre of physicians, nurses, people involved in the medical field that understand the potential serious adverse events that are a little different from what you may see, and we've learned that with chimeric antigen receptor T-cell therapy. So we need inpatient teams as well as outpatient teams who understand that the serious adverse events that evolve over time, and that we need to know how to respond to them quickly.
In your response, that makes sense, but you didn't mention company, so it gets back to my company question. You just described a paradigm which doesn't require
It does.
pharmaceutical.
Actually, it does. So confidentiality agreements, clinical trial agreements, budgeting, that is often what slows the clinical trials process or being able to make these treatments accessible to patients. However boring and non-glamorous that may sound, that is the actual reason that a lot of people don't get therapy. So in terms of when we do bone marrow transplantation and you have rates for care that are basically bundled payments, that helps patients to be able to get access to that therapy. But when you have a complex technology, such as CAR T-cell therapy or stem cell gene therapy, that essentially needs to be done in industry so it's absolutely reproducible, definable, and safe for patients. And that GMP process is something that has been done very well in industry. It's not to say it can't be done in academia, certainly we'd like to do that, but in terms of making it accessible to a broader range of patients, as Dr. Biffy was alluding to, the idea that it goes through industry, it has the process of being refined, and where the technologies have been brought to bear that ensure that there aren't off-target effects, I think that will be done more effectively in industry and then administered to us. So the reason that this has to be done in centers where we know how to do this is because we have the package put together. And that means that our institutional review boards know what to look for when we're talking about serious adverse events. When you think about severe cytokine release syndrome with CAR T-cell therapy, that was just the first thing we knew about for CAR T-cell therapy. Subsequently, there was encephalopathy that led to a few deaths in actually 2 of the trials. So it means that we have to be more educated about patient care in this setting, and that's how we avoid potential adverse events. So it's a real marriage between companies and academia in terms of how we administer these trials, but it means you have to have a designated center of excellence for this, and hopefully a network.
And I think we haven't mentioned it, but sustainability of the system is also relevant, financially speaking. So the cost of these treatments, whatever we are talking about in vivo, ex vivo gene therapy or genome editing, is huge. And partnership with industry could allow us addressing this aspect and increasing the accessibility to the treatment for the patients.
So it's an economy of scale. I think that's really
So,
important.
so let me come to one final issue, which I think is also something which we have to consider. So we heard AAV is really an astounding vector to target almost specifically certain cell types. But I think there's a serious downside to this, that a large majority of people have antibodies against this virus. And one injection, of course, will make antibodies. If you don't have antibodies, one injection, you never get a second one because it will never work. So maybe we should address that. This is an issue which is very efficient, particularly experimentally, but how well is it already to use in the clinic? Maybe anyone of you can address that issue.
Yeah, I think the component is also very related to the disease of interest, target organ, and so therefore the indication, the age at which treatment is administered. So clearly there are tissues like the brain where the need for a second dosing may be less relevant. Others, like the liver, where there could be the need, also based on the efficiency of gene transfer realized by the vectors. Having accessibility to multiple serotypes with similar properties could allow addressing this component as well. And so having left the ability for the same patient to be redosed in case therapeutic benefit would need to be relaunched by a second dosing.
Any other questions to be raised?